Hello guys, this is Dr. Possibility from Excel Academy. Kindly make sure that you subscribe to our YouTube channel and also register with us. In this class, I have a simple task of explaining practical number 2 of histology. And during this practical, we are simply looking at the cell and the cellular organelles. When you talk of a cell is just the basic unit of all living organisms, and this cell contains what we are calling the organelles. These organelles have various functions. For example, when you look at the mitochondria, which is also known as the powerhouse, it is responsible for respiratory process. When you talk of the nucleus, which is the governing body of a cell, its major function is just to govern all activities in the cell and to also store genetic materials such as DNA. There are other organelles that we can talk about, such as the endoplasmic reticulum, which is in two types, and we can also look at the Golgi bodies. So when you talk of this practical, our major purpose is to look at these organelles and how these cells appears. Let's quickly look at our first slide. Remember, this is an histology practical. This simply means that we are going to be using slides, and this slide were being viewed using a microscope. Every slide that is in this video was being viewed and recorded using a camera from the microscope. Please make sure that you subscribe to our YouTube channel and make sure that you register with Excel Academy. So in slide number 35, in this slide we are looking at the scalp. The scalp is just the skin which is found on the scalp. So when this tissue was gotten, it was being viewed using a microscope. So this slide is just the slide which is of a scalp, and this slide was being stained using H&E. So in this practical, for this slide, you are required to locate and see the sebaceous glands. So this is the part for this practical which is the area of interest. So in practical number 2, you need to see the sebaceous glands, and the thing that will make you to see this sebaceous gland is by simply looking at its prominent nucleus. The nucleuses are the ones which are making these sebaceous glands to be so prominent and viewable using a light microscope. So in this practical, you need to draw the sebaceous gland, the cells of the sebaceous glands. So you need to pick few cells, 4 to 5 cells, which are adjacent to each other. You pick them and draw them and simply label them. Now, what are you labeling in these cells? One thing you're going to label is the plasma membrane for those adjacent cells, the nucleus, the nucleolus, and also the cytoplasm. Those are the things that you need to draw when you talk of these slides. All right. So after knowing that, after drawing that, please take note that this is the only thing that is needed in this practical. But when you talk of a scalp has other parts, okay, which you need to know by heart. So these parts, you can talk of the hair follicles, you can talk of the pili muscles, and you can talk of the layers of the skin, which is the epidermis, dermis, and the hypodermis. So when you talk of this slide, This is a slide which has been viewed using a light microscope at high power. So some of the content, please don't get me wrong. In this lab, in these slides, the only thing that you need to draw are the what? Are the 4 to 5 adjacent cells. And these cells, you draw them by viewing the what? The sebaceous gland, which has prominent cluster of nucleus. Now, when you view the sebaceous gland, you'll be able to draw the adjacent cells, 4 to 5 of them. And the things that you need to label in those cells are 1, the nucleus, the plasma membrane, the nucleolus, and also the cytoplasm. But like I said, the scalp has other parts, such as the upper layer, which is the what? The epidermis. Then you have the dermis, then you have the hypodermis, and also you have the hair follicle. This is very easy for everyone to understand. But the examiner would also want you to know this part of the hair follicle, which is known as the follicular bulb. Many tutors, many demonstrators will not tell you this, but these are things that you are going to find in the exam, where the lecturer will project this part and then they are going to ask you label part A. And remember, this part is known as the follicular bulb. So as a student who is studying anatomy, you need to make sure that you understand each and every content. Remember, in this practical, you only need to draw the slides, which is the part which is showing the sebaceous glands. Let's quickly look at this. So this is a slide which has been viewed using the what? The high power, okay? And we have used a light microscope. So if you look at this whole part, this is the what? The sebaceous gland. And this sebaceous gland, you need to understand that they have prominent what? Nucleus. So these are the prominent nucleus. And this prominent nucleus, they have the nucleolus inside there. And this nucleolus, you can look at around, you can also see the what? The cytoplasm. And I'm sure this is the what? The cell membrane, okay? So this is the cell membrane. So you need to draw the adjacent cells, like this cell there, that cell there, that cell there, and also this cell there. You draw this adjacent cell, then you are going to show them using the what? You are going to label them. You label the cytoplasm, the plasma membrane, the nucleus, and the nucleolus. Then you're done. We're talking about the cell and its organelles. But remember, in the exam, you'll be required, okay? In the exam, you'll be required to do what? You'll be required to be able to identify these parts. For example, you can be asked to identify this part marked by what? By yellow arrow. So let's say this is A. So if you look at A, like I said, this is the scalp. And the scalp has different contents that you need to understand. So if you look at A is not pointing at what? The sebaceous gland. Instead, it is what? It is pointing at the muscles, which are the erector pili muscles. Okay? And these erector pili muscles, they play a role in homeostasis. The other component which is being pointed at, this is what? The plasma membrane. And this blue arrow is simply pointing at what? The nucleus. So these are the content that you need to know. It's not just about drawing, it's also about you preparing for the exams. And in this case, because your work will be projected, this simply means that you need to be able to label, like the way I have labeled this part. So in the exam, they can project A and they say identify A. They can project blue and they say identify B. They can also project C and say identify C. So like I've said, A is the erector pili muscles, B, it is the nucleus having the nucleolus, C, it is just the what? The cell membrane. And the whole of this slide, in some exams, they will ask you, name the slide. In this case, this is the sebaceous glands. Automatically, the whole of this slide, it is just simply the scalp. So please take note of that and make sure that you understand. So this is also the slide of the scalp. Okay, your work will be projected, so you need to understand. So this is what? This part, these are the sebaceous gland. These are also the sebaceous gland. These are also sebaceous gland. These are also sebaceous gland. And these muscles which are passing are the erector pili muscles. Guys, we need to understand the content. This is more reason why I am emphasizing on you guys to make sure that you register with Excel Academy. Okay, you register with us. We are going to review what you need to know. We understand how these things are carried. We also understand how the exams do what? Come. So make sure that you register with us. We review more so that you'll be able to enjoy your medical career, medical studies properly. Like I said, other content you can talk about the sweat glands there. All these will be needed for you to be able to see and understand. Let me quickly go on the next part. This slide is just the sebaceous gland, and I have 100% explained the content, the parts, the area of interest in practical number 2. In your case, it can be practical number 1, but that's what is supposed to be known. View that, screenshot, examine yourself, see the nucleus, the nuclear membrane, the sebaceous gland, the erector pili muscles. View all those things and be able to know them by heart, because you'll be examined on that. So this is just the scalp, and in this slide we are just projecting the area of interest, which are the sebaceous gland having a cluster of nucleus, and this cluster of nucleus have the cluster of cells, and this cluster of cells has prominent nucleus which has been stained by H and E. All right. Okay. The next slide of interest is the sensory ganglion. The next slide of interest is the what? The sensory ganglion. Okay. So in this sensory ganglion, what are we trying to understand? Okay. What are we trying to look for? Okay. So in this sensory ganglion, you need to examine the large grayish nerve cells. Okay. The large grayish nerve cells. And you also need to look at the nucleuses, okay? The nucleus. And these guys, they also have prominent what? Nucleus, okay? So those things are the things that you need to look at. Now, what are you supposed to draw in this slide? So what you need to draw is, one, you need to draw 3 to 4 nerve cells with their satellite what? Cells. You need to understand that these nerve cells are being surrounded by what? By the satellite cells. And these satellite cells are just trying to support the nerve cells. So let's quickly look at this sensory ganglion. So this is the sensory ganglion, okay? This is the sensory ganglion, and in this case, you need to look at the what? The cell body. So these are the cell bodies. These are the cell bodies, okay? These are the cell bodies. These are the cell bodies of the what? Of the nerve cells. These are the cell bodies. Now, there is what we also need to look at. We need to look at the what? The satellite cells. So which one is the satellite cells? So these are the satellite cells which are stained. The way you can look at the satellite cells, you can easily view them by looking at the what? Them being dark stained. So these are the nucleus of the satellite cells. All those dark stains are just making the satellite cells, which are simply supporting the nuclear the nerve bodies. Okay, the nerve bodies. So this is a sensory ganglion. In most cases, the way this question comes is that they'll ask you identify the slide, and you should be able to say this is the sensory ganglion. Then the next question will be give the reasons. So you're going to find that this is having it's having the presence of the what? The nerve bodies, and the nerve bodies you can see are these nerve bodies, and these nerve bodies are surrounded by what we are calling the satellite cells, which are dark stained. Please make sure that you follow up and you subscribe to our YouTube channel. The next slide is slide 64, and slide 64 is about the liver. Okay, so this liver has been stained by a special stain, which is PAS. Remember, in my other video I explained about staining, and we have different types of stains. A very special stain which I talked about was PAS. Okay, PAS. And this PAS, it is used to stain glycogen molecules, and it normally stains them red. So in this slide, you'll be able to see the liver being stained, and most of these cells are on the one side. So if you can see, these are just glycogen molecule. The red guys, these are glycogen molecules which have been stained by PAS, and then you have these black spots which are just the what? The nucleus. So you need to make sure that you are able to draw few cells from these cells. One important question which you can be asked in the exam is what fixative or what chemical was used to fix this slide. Okay, so the chemical which was used to fix this slide, it is just a special fixative, which is camoise. Okay, camoise. So camoise is the one which was used to preserve the glycogen granules in the liver, so that we'll be able to stain it using PAS. And when this PAS is used, it's going to stain these glycogen granules red, and we are going to see prominent nuclei. In this case, these are just prominent nuclei, and you are able to draw few cells from here as assigned by your demonstrator or tutors. All right, so one thing about this part, when you talk of the exam part, you'll be required to identify the slide. They'll tell you identify these slides. They can tell you what has made you to know that this is the slide. They'll ask you which stain was used. They'll ask you which fixative was used to preserve the glycogen granules. So make sure that you understand that. The next one, it is the slide 1.11. In this slide, you are much interested in identifying the shape of the shape of the what? Of the cell. So we need to understand that according to the shape, the covering epithelial tissue, remember, there are two types of epithelial tissue. You have the glandular epithelial tissue, which is responsible for glands, okay? And these are secretive epithelial tissue. These tissues are just tissues which come from the covering epithelial tissue in other books they will say membrane epithelial tissue. So when membrane epithelial tissues, their cells, when their cells start to undergo mitosis, meaning they will even start increasing in number. As a result, they will start penetrating into the next tissue, which mostly is the connective tissue. So when they proliferate, and then these guys, they multiply in number, they'll either have a duct or not have a duct. As a result, the glandular epithelial tissue are also being classified into endocline and exocline, depending on the presence or absence of the of the duct. This will be explained later on in the few videos that I'm going to upload. So when you talk of the covering epithelial tissue, this covering epithelial tissue, it is being classified into three classes, depending on the shape. So you have the squamous epithelium, you have the columnar epithelium, okay, you have the columnar epithelium, and you also have the what? The cuboidal epithelium, okay. You have the columnar epithelium, you have the squamous. So this is squamous. Make sure that you take note of that. This is squamous, this is columnar, okay, and this is what? Cuboidal. This is cuboidal. And this class of epithelial tissue can either be stratified or can be simple. We have simple or stratified depending on how it is, but all that will be explained in the tutorial video about the epithelial tissue, which I will make. But in this lab, we are interested in viewing the squamous epithelium. Where was this slide gotten? So this slide was gotten from the mouth, which is the buccal cavity. That's where a section of the cheek was gotten and prepared for us to see the squamous epithelium. So this is a slide. This is a slide of simple squamous epithelium, and in this slide you need to be able to draw the cells which are just simple squamous, and you should be able to identify. So you need to draw about 3 to 4 single cells at high power. So this slide is at high power. So we cannot really appreciate using this slide, but we can also appreciate using this other slide. So in this slide, you can see this is a squamous epithelium. This is the squamous epithelium. You can also talk about it having the nucleus there, and this has easily been appreciated. So this is the simple squamous epithelium, and in this practical you need to draw it using your pen and pencil. So you draw it and you label it. All right. The next slide is the last slide in this lesson. The next slide, it is slide 1.34, which is columnar ciliated epithelium. So this columnar ciliated epithelium, I'm sure some epithelial tissue can be ciliated, some can have stidocilia, some can have microvilli or such type, and this will be explained in the later video. So in this case, we are looking at the columnar epithelium, an epithelium which is appearing in that way, and there is a nucleus and it is having some cilia on top of it. So this is just a columnar ciliated epithelium, and in this case we are using slide H1.3. all right, so please make sure that you register with us and make sure that you follow up. So even this slide, you can't easily appreciate the parts, but one important thing is that you can see some blood vessels in it. Yeah, you can see some blood vessels in it, and this one you need to be able to identify it. But unfortunately, these slides cannot help us. Instead, we can use this other slide. So in this slide, we can simply be able to identify some few content which you guys are supposed to know. So one content you can talk about is the goblet cells there. The goblet cells there. You can talk about the cilia. So this is how we were supposed to view these slides, but you can't appreciate it much unless if we use this other slide. So in this slide, this is the columnar ciliated epithelium, and one important content you can talk of, it is the goblet cells. These are just the goblet cells which have been filled up with mucus, because these cells these are unicellular glands. So goblet cells are special cells because they are unicellular glands and they are able to produce mucus. Yeah. Then the other one you can talk about is the cilia. So this is the cilia layer I was talking about. Then the other thing are these basal cells, and then we have the basement membrane, and then we have the capillaries, which we saw here. I'm sure I mentioned about that. These are just capillaries. Okay, these are capillaries, but the original cells, we could not see them using this slide. So those are just capillaries. So this is what is needed or required for you to draw, except the goblet cells. But you need to show the cilia, you need to show the basement membrane. So this basement membrane here, it is the one which is demarcating the epithelial cells from the connective tissue. And then we have a cluster of capillaries which is in the connective tissue, and this has been viewed in the previous video. So make sure that you follow up. Alright, ladies and gentlemen, like always, Excel Academy, we are not going to offer you our services in line with how you are doing it at class. At class, they are teaching you to know what's everything. But here, we are looking at making sure that you know everything, and we help you to know about the future questions, the possible questions in the exam, how histology is said. So like at school, they'll show you the slides, they'll tell you some few content, of which some few content you'll not be told, okay, and then they'll be done. They'll tell you to go and draw, okay, yeah, they'll tell you to go and draw. Some cases it might not even carry some marks, but it's important for you to draw and understand these labels. But I'm not going to go much in details on that. So as we are focused on telling you, making you know, and then after that we'll help you to know how the exam is. This is how our classes are, and this is how they will forever be. So make sure that you register with us, please. Okay, look at that. This is from an exam question. This is from an exam question. Okay, this is 100% from an exam question. Have you seen me projecting or showing you anything of this? The answer is no. And if you are not lucky, you find that even your tutors will not show you this. Will not show you this. But it is there in the histology lab exam. And you'll be asked to do what? One, the question is, name the slides. What is the name of this slide? What is this? Automatically, it is the what? The mitochondria. That's the mitochondria. Then the other thing that you need to understand is labeling these slides. Labeling these parts. So you have A, B, C, and D. So I'm encouraging you all to make sure that you go and research on what is A, what is B, what is C, and what is D. Then you also give two functions of the part marked A. So this is the mitochondria. Okay, this is the mitochondria which has been examined. So you need to be able to know this slide. Very important for exam. And make sure that you know its labels. Okay, I'm giving you an assignment. Comments in the comment section. Give me the answers of A, B, C, and also give me the function of A. Okay, let's quickly look at that. So the first question is, name the tissue. What tissue is this, guys? This is the liver. Remember what I said? Remember what I said? I said you'll be asked to identify the tissue. After you identify the tissue, they'll tell you maybe to give reasons, but in this case, I can see some labels there. So the first thing is state the reason to your answer. Okay, so from here you can tell that this is the liver. Why is it the liver in this case? There are other reasons, but in this case you can see the prominent nucleus. Okay, and you can also see the glycogen granules which have been stained pink by, which has been stained red by PAS. That's all. Name structure A and B. I'm sure you can see structure A here. This is the nucleus, okay, and structure B there, which are just glycogen granules which have been stained red. You see, this is a reason you need to do what? To register with us. In the lab, they won't explain some of these things. They won't review to you how the test is, how the exam is being said. They'll only tell you to draw and label. But we are telling you what you are supposed to know. This is how the exam is. They won't tell you draw. No, they'll tell you to label, to give a reason, and also to be able to identify. Let's quickly look at this other guy here. Look, what is this? I'm sure just from looking, you can see that this is what? These are goblet cells, these are cilia cells, and then this is the plasma membrane. Okay, so the question is, name the type of epithelium. What type of epithelium is this? What type of epithelium is this? Okay, then the next thing you need to know is state where this epithelium is found. State where this epithelium is found. So one thing I've observed is that there are things that you would have seen, but look at how it is projected. Okay, so if you look at this, what type of epithelium is it? Okay, so make sure that you comment in the comment section. Tell me the type of epithelium which is there, state where this epithelium is found, and also be able to label the parts A and B. So this is your assignment. Tell me the type of epithelium, tell me where it is found, tell me also the parts which have been labeled A and B. I'm sure this is B there. So please make sure that you do that. Then you can also talk about this slide. What type of epithelium is this one? Okay, what type of epithelium is this one? I'm sure someone can easily tell that this is the columnar epithelium, which is the ciliated columnar epithelium. Then we also have this epithelium, which is also a columnar epithelium. So this is a simple columnar epithelium, and you should be able to know where it is found. So these are some of the diagrams that you'll be exposed to. These are some of the diagrams that you'll be exposed to and which you have not even viewed during your lab. So these are some of the diagrams that you need to take note of and also make sure that you handle that question which I have posed there. Please comment in the comment section, because this is how your exam is set. Okay, this is a slide which you guys will see in your lab, and the first question is name the slide, of which I'm sure everyone is able to tell that this is the sensory ganglion. Okay, and then state the reason to your answer. You can easily say it as numerous nerve bodies. You can say a lot of nerve bodies, because when you talk of a ganglion has a lot of nerve bodies in it. So in this case, the sensory ganglion is just showing numerous nerve bodies, and these nerve bodies are being surrounded by dark stained satellite cells. So that's the answer there. Okay, the other one we can talk about is name the slides. I'm sure this is a scalp, isn't it? This is a scalp. This is a scalp. Okay, then you can talk of give reasons to your answer. There are a lot of reasons you can give. One, you can talk about the presence of the sebaceous gland. You can talk about the presence of the hair follicles. You can talk about that. Those are just few reasons that you can talk about. The next question is label the parts A and B. So when you talk of A, this part is just the epidermis, ladies and gentlemen, and this part is just the sebaceous glands. So you see, in this practical, you'll be told to draw the cells in the sebaceous gland, but the exam will need you to label such parts. Okay, this is more reason I'm encouraging you guys, subscribe to our YouTube channel, register with us, give us time to show you what you need to know, what you're supposed to give much attention. Then trust me, you enjoy. All right, let me quickly look at any other questions if I have any, but I think I don't have. So please, this is my last quote for you, which is, there will never be any good doctor without you. So please keep pushing. This was your friend, your brother, your mentor, your anything which you can feel like calling me, Doc Possibility from Excel Academy. Remember, we have opened up our new branch at Ridgeway campus, which is just 5 minutes away from Unza Ridgeway campus, and we are also offering our services at main campus. So make sure that you contact